Imagine for a second that you have access to the smartest entity on the planet. I'm talking about a digital brain that has devoured every single medical textbook.
Every biology paper, every patient record.
Exactly. It's read more in a microsecond than you could read in, like, 1,000 lifetimes.
That sounds like the ultimate cheat code for medicine.
Right. And so naturally, you sit down with this digital genius and you ask it the one question that has haunted humanity forever. Can you help me solve aging?
That is the holy grail. I mean, we've made progress on infectious diseases, we're making headway on cancer, but aging. Aging is still this messy, incredibly complex puzzle.
And here we are, it's February 2026. We've got these amazing tools. GPT-5 is out. Google's Gemini 3 Pro is just dazzling everyone. But here's the big question, and it's why we're here today. Do these AIs actually understand biology?
Or are they just incredible mimics?
Yeah.
And that distinction is, well, it's everything. There's a huge canyon between sounding like a doctor because you've memorized the vocabulary and actually understanding the machinery that keeps us alive.
So to figure this out, we are doing a deep dive into a brand new piece of research. It just dropped last week, January 30th, 2026. It's titled LongevityBench. Are SOTA LLMs ready for aging research?
And it comes from a really heavy-hitting team, Alex Zavaronkov and Fedor Galkin at In Silico Medicine and the Buck Institute.
And what they've done is, well, it's just fascinating.
They basically decided these AI models needed a massive reality check. So they built a, quote, gym for science.
I love that metaphor. A gym for AI.
It fits perfectly. They didn't just give these models a written exam or some multiple choice quiz. They threw them into the absolute deep end of biological data.
So this LongevityBench, how big of a workout are we actually talking about here?
Oh, it's massive. We are talking about 30,193 specific prompts.
30,000.
Yep. And in AI terms, that's about 50 million tokens of data.
50 million. Okay, so this is not a pop quiz. This is the bar exam and the MCATs all rolled into one.
And the goal wasn't just to see if the AI could recite facts. It was to answer a much deeper question. Does the model have a coherent internal representation of life?
Can't connect the dots.
Can it connect the dots between a tiny molecule and a person living or dying? Yes.
So let's walk through the equipment in this gym. If I'm GPT-5, what am I actually being asked to do?
So they tested the models across the full spectrum. First, they put them on the treadmill with real clinical records, specifically NANEs for 3 weeks.
That's the National Health and Nutrition Examination Survey, right?
Correct. It's the gold standard. Real people, real blood tests. They gave the AI these records and asked a pretty brutal question. Is this person going to be alive in 10 years?
Wow, okay, high stakes right out of the gate.
Extremely high stakes. But then they moved to the heavyweights genetics.
Right.
They used databases like OpenGenes and Synergy Age. But they didn't just stick to humans. They looked at our favorite lab partners, fruit flies and mice.
Ah, the classic Drosophila. Why flies, though? If we want to solve human aging, why are we quizzing the AI on bugs?
Because flies are basically the speed run of aging research. They live short lives, so we can see generational changes so quickly.
Makes sense.
So the researchers showed the AI genetic mutations in these flies and asked, if we tweak this gene, does the fly live longer or does it die sooner?
So the AI has to predict the consequence of a single mutation.
Exactly. And then it gets even more granular. They tested what's called omics.
Omics. I hear this term thrown around a lot. Break it down for us.
Think of it as the different layers of instructions in your body. You have the DNA, that's the main blueprint.
Right.
Then you have transcriptomics, that's the RNA, the messenger carrying the instructions. And then you have proteomics, the proteins.
And the proteins are the actual workers, aren't they? The little bricklayers.
Exactly. If DNA is the architect and RNA is the foreman, proteins are the ones actually building the house. The researchers threw all of this at the AI. Everything.
It really is the full stack. Okay, so I know everyone listening is dying to know. Who won? Who's the valedictorian?
It was an incredibly tight race, a photo finish, really. But the gold medal went to Google's Gemini 3 Pro.
Gemini. So it took the crown. Was it a blowout?
Not at all. Gemini 3 Pro had an average rank of 5.0, but right on its heels were OpenAI's GPT-5 and o3.
How close?
They were tied at an average rank of 5.06.
That is razor thin. We're talking decimal points.
So it's effectively a three-way tie for dominance. But, and this is a really big but, you can't just look at that average score.
Why not? I mean, if Gemini won, it's the best, isn't it?
Not necessarily. If you dig into the data, the winner really depends on what you're trying to do. And the authors highlight this perfectly with Claude Sonnet 4.5.
Okay, where did Claude land?
On average, it was mediocre, not leading the pack. But in one specific event, cancer survival prediction, Claude Sonnet 4.5, didn't just win, it destroyed the competition.
Really? So the overall average student is actually a secret genius at oncology.
It got nearly 70% accuracy in predicting which cancer patient would live longer just based on their tumor profile. The other models couldn't even touch it in that niche.
That is fascinating. It's like a student who gets C's in most subjects, but is an absolute prodigy in calculus.
Exactly. And that's the first big lesson for you listening. There is no God model yet. If you're a cancer researcher, you don't care about Gemini's average score. You want Claude. You have to pick the right tool for the job.
So we have a leaderboard, but it's more like a team of specialists.
Yeah.
But I want to get into the part of the paper that, honestly, kind of made me scratch my head. This idea of an illusion of understanding.
Yes, this is one of the most critical findings, I think.
What does that even mean?
It means the models are incredibly sensitive to how you ask the question. It's a huge tell.
Okay, give me an example. Because if I understand something like, if I get that 2 plus 2 is 4, it shouldn't matter if you ask me what's 2 plus 2 or is 2 plus 2 equal to 5. I should get it right either way.
Logic would dictate that, yes. But let's look at the methylation aging task. This is where they look at DNA markers to predict how old a sample is. When they asked Claude Sonnet 4.5 to guess the specific age group, a multi-class task, it was fantastic. Nailed the age brackets.
So Claude knows how old the DNA is.
You would assume so. But then they just switched the format. They gave Claude 2 samples and asked which one is older. A simple pairwise comparison, and Claude failed significantly.
Wait, hang on. That makes no sense. If I know person A is 80 and person B is 20, I automatically know person A is older. How can it know the age but fail the comparison?
That is the paradox, and here's the kicker. The Gemini models were the exact opposite.
What do you mean?
They were brilliant at the comparison. Sample A is older than sample B. But when asked to predict the specific age number, they dropped right to the bottom of the leaderboard.
That is just bizarre. It suggests they aren't actually thinking about biology at all.
Precisely. It suggests they're relying on statistical correlations tied to the format of the question, rather than a coherent internal model of biology.
It's like a parrot that can say Polly wants a cracker, but doesn't actually know what a cracker is.
That's a fair analogy. And the researchers dug even deeper into this with what they called the context experiment.
This was with the fruit fly mutations, right?
Yes, the multi-mutant lifespan task. They wanted to see if giving the AI more background info would help it make better predictions.
Which, intuitively, it should. If you let me use the textbook during a test, I'm probably going to do better.
And for most models, that was true. In the minimal version, they just gave the mutation names. In the full version, they added gene ontology data, basically the textbook definition. Gemini 3 Pro and GPT 4.1 got much smarter. Their accuracy jumped. They read the material and applied it.
Good students.
But then there was the outlier, O3.
The OpenAI reasoning model.
Yes. O3 didn't improve. It performed almost exactly the same, whether it had the minimal info or the full textbook description.
So hold on. If giving it more information usually helps, why would O3 be the exception? That seems counterintuitive.
That's what you'd think. But the hypothesis from the researchers is really interesting. They think O3 might have already internalized that external knowledge during its training.
So it memorized the book already?
Essentially, yes.
Yeah.
It didn't need the prompt to tell it what the gene did because it already knew. It suggests O3 has a different kind of memory or training depth. It's the student who doesn't need the cheat sheet.
I want to pivot now to what I think is maybe the most disturbing finding in this paper. We talked about how these models are smart, but are they optimistic? Because the paper suggests they are massive pessimists.
Oh, profoundly pessimistic. This came out in the general population survival task.
Right. Back to the epigenetic data predicting how long a person is left to live.
Right. And almost across the board, the models systematically underestimated survival. They compressed everyone's predictions into the 50 to 100-month range.
50 to 100 months, that's what, 4 to 8 years? That's it.
That's it. Even for people who actually lived for another 250 months, that's over 20 years. The AI was predicting they'd be dead in 6 or 7.
That is actually terrifying.
Yeah.
You're saying if I feed my perfectly healthy record into this, it might tell me I've got 6 years to live. Why are they such doomers?
The authors propose a very smart theory, and it goes back to the training data. Think about the text these models learned from. Medical literature, case studies.
Okay.
In those texts, when words like disease or diabetes or hospital appear, what usually follows?
I guess. Bad news. Complications. or death.
Exactly. Death, failure, decline. The success stories of people who manage their diabetes for 40 years don't generate thousands of pages of urgent medical text. They just live.
So the data is skewed.
Yeah.
The AI thinks every diagnosis is a death sentence because it reads more obituaries than lived happily ever after stories.
It creates a pessimistic doctor, AI. It sees a risk factor and assumes the worst-case scenario because statistically that's what it has read about most.
That is a massive, massive flaw. Imagine using this in a clinic. You can't have an AI telling manageable patients to get their affairs in order.
And it highlights why benchmarking like this is so critical. If you just chatted with the bot, it would sound very authoritative. Based on elevated glucose, estimated survival is 60 months. Sounds official, but it's just wrong.
It's hallucinating doom. Let's talk about the other area where the model's just completely face-planted. We talked about DNA, RNA, proteins. The paper calls this the generative gap.
This is the frontier. We aren't just asking it to predict an outcome, we're asking it to fill in the blanks. Here are 50 genes, predict what the other 50 are doing.
Like autocomplete, but for your biology.
Exactly. For transcriptomics, the RNA top models like Gemini 3 Pro did okay. They had a Jacquard similarity score of about 0.176.
Okay, hold on. 0.176. To the average listener, that sounds low. Is that a failing grade?
In school terms, yeah, it's a failing grade. A perfect score is 1.0. So we're talking about a student who is getting maybe 20% of the answers right.
Okay, so even the good result is barely passing.
It's better than random, which would be near zero. But here's the drop-off. When asked to do the same for proteomics, the proteins, they failed miserably.
How bad is miserably?
The best score was 0.030.
0.03. That's essentially zero.
It's noise. It's 6 times worse than their already not great performance on RNA.
But why?
Yeah.
Why is protein so much harder for them than RNA?
2 big reasons. First, data scarcity. We have oceans of RNA data. Proteomics data, especially the high-quality Olink data they used, is just newer and rarer.
So the models haven't read enough about it. And the second reason?
The second is that biology is tricky. The AI might be assuming that protein levels match RNA levels, and that's just not true.
Okay, give me an analogy.
Think of RNA like an IKEA instruction manual. It tells you how to build a bookshelf. The protein is the actual wooden bookshelf in your living room.
Okay, I'm with you.
Just because you have the RNA on your table doesn't mean you have the finished bookshelf. You might lose the screws or just get tired and throw the manual away.
Or you might just be lazy and never build it.
Exactly. That's called post-transcriptional regulation. The AI sees the manual and assumes, oh, there must be a bookshelf. But biology doesn't work that way. The AI is making a logical jump that reality just doesn't support.
That makes total sense. The AI is too literal. It thinks the map is the territory.
And that's a nuance you only learn by looking at the actual data, which the models clearly haven't mastered yet.
So let's zoom out. We've got Gemini barely winning. We've got a specialized savant in Claude, this really scary pessimistic streak, and a total failure in understanding proteins. If I'm a researcher or just someone listening to this, what's the takeaway? Can I trust these tools?
The verdict from the team is cautious. It's trust but verify, and in some cases, it's just do not use yet.
Let's break that down. Clinical records.
They're useful assistants, but they're highly pessimistic. Do not take their survival estimates literally. Treat them as a worst-case scenario generator.
In proteomics.
Don't use them. Not yet. The capability simply isn't there. If you try to generate protein data with GPT-5 today, you are effectively generating hallucinations.
But the paper isn't just a critique, right? It's called a gym for a reason. Gyms are for getting stronger.
That's the most exciting part. The purpose of LongevityBench isn't just to grade the models, it's to train them. They propose a feedback loop.
A training montage for the AI. Cue the Rocky music.
Exactly. They show this concept called the MMAI gym. You take these 30,000 prompts and you use them for reinforcement fine-tuning. You force the model to take the test, fail, correct itself, and learn the actual biological ground truth.
So we use the benchmark to beat the pessimism out of the model. We force it to realize the IKEA manual isn't the bookshelf.
And if we do that, we move from chatbots that know biology terms to what the authors call biological superintelligence, an AI that actually aligns with the reality of how life works.
That's a dream, isn't it? An AI that doesn't just mimic a doctor, but can actually see patterns that we can't.
We are not there yet. This paper is a reality check, but it's also a roadmap. It tells us exactly where the potholes are so we can fix them.
It really changes how I look at these tools. You know, we all get dazzled by how good they are at writing emails or code. But biology. Biology is the ultimate black box.
And right now, the AIs are just reading the label on the box instead of looking inside. This benchmark forces them to open the lid.
Before we go, I want to circle back to that pessimism bias one last time. It's really stuck with me.
It is the most provocative takeaway for me as well.
If these models are learning to be pessimistic doctors because of the text they read, what other biases are hiding in there that we haven't even found yet?
That is the question we have to ask.
I mean, think about the implications. If they're biased about death, are they biased about treatments? Are they biased towards pills over lifestyle changes because pills just appear more often in medical texts?
Or think about insurance. If we rush to deploy these pessimistic doctors, are we going to see insurance companies using these algorithms to deny long-term care? Because the bot says the patient won't live that long anyway.
That's a chilling thought. The stakes are so much higher than just a wrong answer on a test.
And we haven't benchmarked those biases yet. Until we do, we're flying blind.
A sobering thought to end on, but a really important one. We're at the beginning of research-grade biological AI, but it sounds like we have a lot of work to do in the gym.
Absolutely. The potential is infinite, but the validation has to be rigorous.
For those of you who want to see who is currently winning this race, because these models update constantly, the authors are keeping a live leaderboard up. You can check it out at bench.insilico.com.
It's going to be a fascinating race to watch.
Thanks for diving in with us. We'll see you in the gym.